Hello learners, welcoming to this session. Myself Dr. Suresh Kumar, Scientist C from National Institute of Cancer Prevention and Research, Indian Council of Medical Research, Noida. I am welcoming you all for this polymerase chain reaction lecture series and a postgraduate diploma in environmental occupational health. Let's go about polymerase chain reaction. Before going to that, let's see the basics. If you see that animal cell structure, you have an outer membrane, plasma membrane, and inside you have cell organelles, and inside you have nucleus. So inside nucleus you have genetic material which is made up of DNA and RNA, and also you have this whole cellular plasma that contains proteins. So before development of PCR, there are important discoveries that laid the platform for development of PCR. Important discoveries are DNA structure determination by Watson and Crick, and discovery of DNA polymerase enzyme that is called DNA polymerase by Arthur Kornberg. He is the one who discovered that during a replication, in the sense that copying of parental DNA into daughter DNA by DNA polymerase enzyme. So this is a major discovery in this development of PCR. Then interpretation of genetic code by Hargobind Khorana, Indian origin Nobel laureate, who has discovered triple codon by artificial synthesis of a gene. In Dr. Khorana's lab, there was a student Kleppe who left that message that by artificial synthesis of genes is possible, and this can be accomplished by using the enzymes which is called DNA polymerase. So these are the important discoveries, and Thomas Brock and Hudson Fries, who during the same time they have identified Thermus aquaticus from they identified DNA polymerase enzyme from Thermus aquaticus, which does the same DNA polymerase at the higher temperatures. So the DNA enzyme polymerase isolated from Thermus aquaticus, and it can work at higher temperature. So these development actually merged together for the development of in vitro replication called PCR. So the first paper on PCR appeared in 1985 in the Science paper. So what is a PCR? So what the PCR does? PCR is polymerase chain reaction. That is polymer, basically a polymer is made by ASE enzyme, that is polymerase. So monomer become dimer, dimer become trimer, and become tetramer, and become oligomer, then become polymer. This polymerization reaction is being done by enzyme called polymerase. So this is having a DNA as a template. This is DNA dependent DNA polymerase. So the PCR technique, basically the discovery of PCR was done by KARRY MULLIS. About his biography, he has worked on ion transporter during his PhD, and he did his PhD in 1979, and after that there was no job, and he was working in bakery. And he joined in 1981 in the CETUS Corporation as a technician. So during this time, he was involved in the artificial synthesis of oligonucleotides, and he invented this polymerase chain reaction along with other colleagues. And with a lot of disputes, finally his name is announced for Nobel Prize. But however, that KARRY MULLIS have not actively participated after in the academic research. The Psyche, Mullis, Ehrlich and the CETUS Corporation together, they published their first paper in 1985. So this is the start of PCR. From that onwards, that PCR has got enormous growth in multiple ways and multiple formats. So as Watson Crick has discovered, DNA is a double stranded and it is antiparallel, and it has 4 nucleotides: adenine, guanine, thymine, cytosine. And adenine pairs with thymine and with a hydrogen bond, and guanine with cytosine. So this is the basic structure. This is the DNA, and how the nucleotides are having the phosphate, sugar, and nitrogenous base. And this is purine pyrimidine. This purine has adenine and guanine. Pyrimidine is cytosine and thymine, and adenine pairs with thymine, and guanine pairs with cytosine. So polymerase chain reaction. Polymerase chain reaction basically consists of 3 steps, which was the DNA is first of all denatured into single stranded. So once single stranded, you can have a primer, a piece of DNA which can be targeted to a piece of DNA, and have a forward primer and reverse primer. This forward primer and reverse primer can work, right. So once a primer has been annealed with the target, that has been extended by DNA polymerase, that is Taq polymerase, and this is once the copy is made, the same copy can act as a template in subsequent amplification reaction. So the extension again goes back to the previous cycles of denaturation of DNA. So this become a cycle. So this cycle keep on going on. So this is why it is called polymerase chain reaction. Yeah. So this polymerase chain reaction that consists of three steps: denaturation, annealing, and extension. This all the process has to be accomplished at the particular temperature profiles. So to accomplish denaturation, we give a temperature with a high temperature, that is 95 degrees C. So that temperature, this double stranded DNA become a single stranded. So next step is annealing. The primer has to be attached with the complementary sequence, that is called annealing, and this process has to be accomplished in particular temperature. It's a permissible temperature for annealing, that is 50 to 60 degree. And once this annealing has been done, this primer is being extended by Taq polymerase. That extension has to be done at 72 degree. So these are the three temperature profiles has to be followed, and this is a reaction keep going on back and forth. So amplification type is linear amplification and another is exponential amplification. The one which we see in the PCR is exponential amplification, but however in PCR we can utilize a linear amplification also. This linear amplification, if you give if you give 10 copies, you will get a 10 copies of amplicons. But whereas in the exponential amplification, it is a geometric amplification. If you give 1 copy, after 30 cycles you will give 2 to the power of 30 copies. So this is exponential amplification. The typical PCR reaction components: DNA template 50 to 100 nanogram and forward primer 0.1 to 0.5 micromolar or 2.5 to 12.5 picomolar, and DNTP is 50 to 200 micromolar. Buffer is 10x, is supplied as a 10x, and you have to give 1x. Taq polymerase, you have to use 0.3 units, and distilled water, it has to be final volume to the 25 microliter. This is the typical PCR reaction component. So a good template, as we see that in the next, we saw that 50 to 100 nanogram template. Template is any genomic material, that is maybe a DNA or RNA that needs to be amplified, considered as a template on which this all the process has to happen. So this is the template is a genetic material on which the primer, polymerase and DNTPs all sit together and they extends and make a copy. So the template acts as a permanent reference based on which all the pieces of DNA material or amplicons will be made. If it is an RNA, it needs to be made as a cDNA copy, that is called reverse transcription. So the RNA needs to be reverse transcribed by reverse transcriptase enzyme. Once the cDNA is made, then the usual process of PCR will be carried out. So any gene to be studied or cloned or RNA material to be RNA is to be preferred for the PCR. If I want to express a gene, then we look for a mRNA which has been transcribed, and this mRNA has been made as cDNA and that has been cloned and it will be used for expression studies. If for any mutation studies for genetic diseases, we use a DNA material, and for tissue specific expression, RNA is considered from tissues, and for mutation, DNA is preferred. Primer. Primer is a short piece of DNA which is 18 to 20 bases in length, that is designed against particular target. You want to amplify a particular region of DNA, so you have to design your primer which will be a complementary to the bases where you want to amplify. So the primer can be designed by there are software sites are there which is freely available. One can easily adventure that's Primer 3 software or NCBI primer designing software there, which is free, and this is academically this primer softwares are used more in most of the academic settings. So primers generally 15 to 30 bases long. So ideally we take 18 to 20 bases, and important parameters is that the primer, both the primer, forward primer, reverse primer, they should not have complementary of each. Then there should not be any homology of this primer sequences with any other gene other than your target. So that is very important. So it should be a unique, your primer binding site has to be unique to your target gene. And second thing, no self loops. You can see that the primer itself should not bend together and forms a self loop. So these are the parameters one has to consider at the time of primer designing. And the primers, both the primers should have equal melting temperature for assessing the annealing temperature. So DNTPs, all the DNTPs you have in equimolar concentration, adenine, guanine, thymine, cytosine, all has to be in equimolar concentration. And you see that the PCR amplicons will be done in from 5' to 3' direction. Buffer components, buffer basically tris-based buffer, ammonium-based buffers. This tris-based buffers generally that helps in stabilizing the pH. When you increasing the temperature in the PCR reaction, the tris buffers actually stabilize the pH. And Taq polymerase actually activate maximally at 50 millimolar KCl, and also MgCl2 also needed for activity of Taq polymerase. So generally we use 1.5 millimolar MgCl2, but however when you don't have any PCR products, we generally increase the MgCl2 to 2.5 millimolar MgCl2. So this will help you for the Taq polymerase activity. So MgCl2 is a cofactor for Taq polymerase activity, and this has been normally used in most of the PCR applications. And Taq polymerase, this has been isolated from Thermus aquaticus, and this enzyme works at higher temperature. It polymerases nucleotides at 72 degree, but now there is a variant of polymerase has come. They can act as even a 60 degree C. So you don't have a separate amplification setup at 72. The annealing part as well, annealing as well extension works at 60 degrees. So most of the real-time PCR applications has carried out 60 degrees C. So there are different types of PCR technique: conventional PCR, reverse transcription PCR, real-time PCR, nested PCR, multiplex PCR. We'll just see few things. So nested PCR, if you want to really have a huge amount of template, and if you want to do allele specific some studies, so you will actually amplify your DNA in the outside of your target, then you have a different PCR primer for inside. So outside and inside, this is a nest basically. So this is called nested PCR. So you will perform first outside PCR. From outside PCR, you perform inside PCR. This is two primers you have to use in this nested PCR. So this is asymmetric PCR. Asymmetric PCR is not used most of the applications, but it is for some, if you want to do some biosensor applications, if you want to do sequencing reaction or probe preparations, they use asymmetric PCR, in which that forward primer will be more and reverse primer will be less. So 1:10 ratio primer combination is used. So touchdown PCR, sometimes your target is not amplified. To really do that, they use different temperature profiles. So it is used generally for hard to amplify target. So they decrease the annealing temperature for every cycle at 1 degree or 0.5 degree of amplitude. Then they look for after 15 cycles, if you got amplification, then they go for a normal PCR cycles. Multiplex PCR. Multiplex PCR is in a single reaction tube. You want to do multiple gene amplifications. That is always possible. You can put together multiple gene primers. You can do 8 pairs of primer, 10 pairs of primers. People have done up to 21 pairs of primers targeting a different gene target regions, and you can amplify that. And this is a gel which shows that multiple genes. So in a single PCR tube, you can do multiple primers to amplify multiple targets. This is a PCR RFLP. If you want to do SNP studies, you can have a PCR of this particular region, and you can do a restriction digestion, or you can use a primer which is specific for your SNP. So if you have variation in that particular, that PCR will be amplified. So the primer designing can be done in this, was specific for only for particular variant or wild type. So that is also possible. So arbitrary primed PCR. This is one of the biggest application in where there is no prior sequence information is available. If you go to anywhere, like if you go to moon, how will you identify a microbe? So for that, arbitrary primed PCR is used. You don't have a primer sequence defined here. The primer sequence is not designed, and these are random primers. So the random primers amplify all the DNA sequences, and that's been amplified. This is called arbitrary primed PCR. So one can easily amplify any DNA. And this is methylation specific PCR. Like if you want to study a methylation pattern in the gene, So you can bisulfate treatment, you can give a bisulfate treatment, and only methylated DNA and unmethylated DNA can be used by using different primers specific for unmethylated, methylated. Coming for reverse transcriptase PCR. Reverse transcriptase PCR is that when you want to do RNA material, if you want to amplify the RNA material, first of all RNA material has to be reverse transcribed. So here you use RNA dependent DNA polymerase. So this is a reverse transcription reaction in which that mRNA is taken and primer can be poly-T. mRNA has a characteristic poly A tail at the end. So you can use a poly-T primer to make a cDNA copy. So once the cDNA is made, then you can use, so you will have a pool of mRNA which amplify all the mRNAs. Now you can use gene specific primers and taking the cDNA as a template, and you can set up a normal PCR. So in this way you can clone any gene, any protein coding gene or multiple protein coding by keeping the cDNA as a template. And also that can be used as for copy number valuation by real-time PCR. Real-time PCR, this is one of the most needed application now, everybody should know now. So real-time PCR is basically a PCR, but you will be monitored in real time. So this is an amplification of target. Basically in normal conventional PCR, we do PCR, then we will take it the PCR reaction to the gel, and we analyze in the agarose gel and look for then whether the PCR product is or not. But real time PCR, what it does is that the amplification is done and it's been monitored in real time. And important aspect is that you can quantitate a copy number. So that is one of the main thing. Like most of the applications, like in the genetic diseases, you have over amplification, over expression, what we call over expression, over amplification. So this you can identify by real time PCR. So the computer screen which is attached with this real time PCR machine, that shows cycle by cycle the reaction, how the amplification that happens. The PCR amplification can be used as a semi-quantitative or quantitative or qualitative. The copy number of template can be deduced from measuring amplification cycle. So let us see how it is. So basically real-time PCR use, mainly most of the people they use cybergreen based, dye based real-time PCR or TACMAN based oligo probes which they will use. So the dye based, I will see that what is dye based. So real-time PCR you have two types, absolute quantitation and relative quantitation. Absolute quantitation is that you have to clone a gene and put it in a plasmid, and now you assess the copy of this plasmid, then you put up a 5 different concentration, then you do a PCR, like then you make a standard graph. So that way it is absolute quantitation. Relative quantitation is that you are quantitating your target gene with a housekeeping gene. So the ratio of this housekeeping gene to your target gene, that is relative quantitation. So this shows that this much fold increase or decrease with respect to the housekeeping gene. So housekeeping gene actually generally that does not alter its expression by any kind of environmental factors. So real-time PCR reaction, this is cybergreen based. So basically this is what PCR reaction, you know that this is a normal PCR reaction, but whereas that you have stars here, the stars is the dye. This is basically intercalating dye. The cybergreen binds with the double stranded DNA and the fluorescent emitted is proportional to the copy number. Actually once the cybergreen, the free cybergreen fluoresces very less, but the bound one which fluoresces tenfold increase once it is bound with the DNA. So the amount of signal is proportional to the copy number. Real-time PCR by TACMAN probe. If you see that here, the TACMAN probe basically a fluorescent molecule. Basically you have primer, in addition to the primer, in between the primer binding site you have a probe. This probe has a different fluorescent formats, fluorescent colors. This probe called reporter and quencher. R is a reporter and Q is quencher. When this reporter and quencher is together, the quencher actually stops all the signal emitted by reporter dye. So when it simply binds with the target, it won't emit any fluorescent. But when you have a PCR amplified, the Taq polymerase basically have that exonuclease activity that chew up this probe. When it chew up probe, it releases a reporter and quencher from the target. So now the reporter gives out the signal. That signal is being measured in the PCR machine. So the signal emitted by reporter is proportional to the copy number of target genes. So the reporter is basically different reporters comes, FAM, hex and many more. Generally FAM and hex has been used, Tamara dye. So there are a lot of dyes are there which can be used for a reporter as a reporter dye. So important thing is that so you can do multiplexing also real time PCR. You can in the single DNA material you can use FAM dye and hex dye for a different reaction, and you can measure the gene expression of multiple genes in a single time in a single tube. So particularly I am coming to this is an important application. So in real time PCR what happens that the PCR amplification is being monitored on online, I mean on online. Thing is that after 10-15 cycles till 10-15 cycles you won't get any signals in the screen, but after 15-20 cycles you will find that there is a graph that emerging out from the baseline. Actually the cycle at which the graph arises above the threshold, that is called CT value. So this CT value is basically referred for measuring the copy number. This is very important. So you have different phases in this graph, exponential graph and linear graph and log phase, and you have a plateau phase. So you have to measure in the growing phase, any phase, and any cycle which is just above the threshold baseline is considered for CT value measurement. Okay, I'll tell about the COVID-19 testing by real-time PCR by multiplex PCR. You have the human cell in which is COVID SARS-2 infected human cell. You have viral RNA as well, human RNAs are there. So here in this side that you have a viral RNA structure, you have a ORF1ab and SEM and genes, which is a 29 kb in total length. And you can have a primers for ORF1ab and N gene and as well RNSP from the human. So these three genes together you can put as a multiplex PCR. Once nasopharyngeal samples have been taken and RNA is extracted, and once RNA is extracted the reverse transcription will be happen, and the real time PCR with all three primers like N gene, RDRP and human gene RNSP. You can see the graph which came out from the PCR machine. You have multiple graphs here. You have CT value of graph which is 15 or 20, 25 something, and it also 40. So here the different graphs are here. The lower the CT value is, the higher the copy number, in the sense that 1000 copy gene will show up amplification at the earlier cycle. The 10 copy cycle will come later on. So basically the lower the CT, higher the copy number here. So the 40 CT value what we have here we consider as a negative because a single copy of gene can give a signal at 34 cycle CT value, single copy at 100% efficiency. But however 100% efficiency doesn't happen in the PCR. Assume that 90% efficiency will happen. So we keep the threshold of considering a positive up to 38 cycle. Any signal that comes up to 38 cycle considers in a positive. So in this graph you can see that the last cycle which we consider as a negative, basically that is a CT values of 40. 40 value is negative because it is a basal background of dye fluorescence. So the lesser the CT value, the higher copy number. So this has been told in most of the COVID testing laboratories. What is the CT value? CT value is 20 CT to 30 CT, 40 CT. 20 CT is that higher copy number. 40 CT or 35 CT is that borderline CT number. 40 is negative more or less. And here we have three genes, N gene, RDRP, RNSP. If you consider all three genes amplified, that is positive, or any one gene of target gene, RDRP or N gene is amplified, then also it is positive. Both the genes of target, RDRP or N gene is not amplified, or only RNSP is present, that means negative, in the sense that your cell is not infected with this thing. So after 14 days of your COVID infection, you will see the clearance of a viral gene. Basically you will find this kind of thing. You won't find any N gene or RDRP gene, but you will find RNSP gene, which is a human RNA. And if you find on all no amplification, that means that your sample is not having proper RNA, then you have to go for resampling. So this is the major application of real-time PCR. In the recent days, it become a big for everyone knows about what is real-time PCR now. So there is application, lot of applications. So it has been found in most of the diagnostics, bacteria, virus, helminthes, protozoa. You can identify aerosol spreading. You can identify by the PCR. It has been used for diagnosis of genetic diseases, mutation, single nucleotide polymorphism. You can identify differential expression of genes. Nowadays, people use a precision medicine. This is being used for this kind of differential expression of genes. Food adulterations, a lot of meat, in case of meat industry, they mix up meats, so people can identify by cytochrome B RFLP, which is specific for particular meats. And also forensic, you can identification of accused in crime scenes, you can identify by based on the DNA fingerprinting. In forensics has a lot of applications. In anthropology, like identification of primitive creatures from the human dwellings, you can identify by PCR reactions. And still there are a lot of modulations, many more applications that the PCR has different formats. And so these are the different again applications, like you can identify the microbiome. If you don't have any knowledge about what is the microbes, you can just take a mud and you can do this PCR, cysteine RNA PCR. You can identify which microbes are dwelling in this particular. So you don't need to isolate or culture this bacteria. So without culture, you can identify microbes in this PCR. So there are a lot of applications. So in summary, PCR has enormous potential applications in various fields in medicine, agriculture, environment, food safety. And PCR application has revolutionized the whole genome sequencing methods. It eases many of the discoveries, including isolation, identifying microorganisms in moon or in the planets. One can improvise this technique according to the needs, and no molecular biology can exist without PCR. With that, thanks.